All right. I would like to thank to Susie to have us present here on our work on susceptibility MRI for doing mesoscale imaging to assess white matter connections and myelin structures. So we know the tissue magnetic susceptibility can be an important MRI image contrast to reveal important information about the tissue composition and also microstructure. So usually we use a gradient echo sequence with phase reconstructions, and we can calculate the so-called quantitative susceptibility mapping, which can give kind of unique contrast in those iron-rich structures and also the cortical rebounds. And if we have multiple data sets like this, we can also extend the tissue susceptibility model into a higher order tensor model and to do susceptibility tensor imaging. So the project I'm going to talk about briefly today is about high resolution susceptibility tensor imaging, aiming at imaging the white matter connections and the myelin structures. Since the susceptibility and anisotropy has been observed many years in those myelinated white matter fibers. Of course, the susceptibility MRI may have some potential advantages as compared to diffusion MRI, that it can easily achieve a high resolution, especially at ultra high field. But it also has a lot of challenges, as I will talk about shortly. So over the years, people have shown that the susceptibility tensor imaging can be a useful tool to investigate tissue microstructures, especially ex vivo. And this can be applied not only in the white matters in the brain, but also in like muscle fibers in the heart, and also those collagen fibers in the knee joint. But doing STI in vivo is quite challenging, partly due to the limited orientation sampling, especially in the tight head coils in our MRI scanner. So ideally, we want to sample a wide range of orientation sampling. So as compared to diffusion MRI, we apply the diffusion gradient for susceptibility tensor imaging. We'll have to rotate the imaging object at different orientations with respect to the magnetic field. So as you can imagine, it's time-consuming, not kind of comfort for the participants, and also the rotation range is quite limited by the coil space. And over the years, people have proposed different strategies to do the regularization for STI. But overall, the performance still not that good in the sense that the reconstructed susceptibility tensor is not coherent enough to do like a whole brain fiber tracking. So to address this challenge, our group has proposed a deep learning-based reconstruction for STI. Basically, we used a physics-informed convolutional neural network to solve a regularized STI reconstruction. And the neural network was trained to learn the important priorities and serves as a data-driven regularizer. And the training data was generated by creating some brain phantoms using in vivo multi-orientation QSM and also the corresponding diffusion tensor imaging data. And also the field image was simulated using the known STI forward model. And here just showing some example reconstruction results using the deep STI that we acquired in vivo. So as compared to conventional STI approach, the deep STI can give a much more complete tracking of the fibers over the whole brain, and also in specific fibers like those going across the corpus callosum. But of course, for this current deep STI network, it has only been trained with data at about 1 millimeter isotropic resolution. So it does not yet generalize well to high resolution imaging yet. So to facilitate high resolution STI, we first developed a STI protocol to scan post-mortem human brain samples at 7 Tesla. So we developed a customized 3D container that allow us to do STI imaging at 7 Tesla at our human scanner. And here just showing some examples that we acquired at 12 different orientations, showing large orientation dependence. And using this high resolution data, we can reconstruct pretty high quality QSM, which allows us to visualize a lot of anatomical substructures, such like the line of January and also the layer 2 islands in the endorenal cortex. Of course, for a better training of our deep STI network, we also want to acquire high quality diffusion MR data. So that's why we last year we came to SUSE and asked for help to get this high resolution diffusion MR data that can be scanned on the same human brain samples we have. So for this, we acquired a 0.55 millimeter isotropic resolution with segmented 3D EPI. Also with multi-shells, and the image quality looked really nice. And for in vivo scans, we also leveraged the advanced Roma EPTI sequence. We acquired the corresponding 0.5 millimeter isotropic resolution for in one healthy volunteers. While we are still working on our STI reconstruction and creating new training data sets, so Yuto and Kenichi first did some initial analysis on this high-resolution data matched with both susceptibility and diffusion data using an atlas-based approach. We also generated some cortical surface map to visualize the diffusion FA and QSM in different cortical regions. And from these maps, we see some interesting cortical contrast patterns. For example, in the precentral areas, we see both high elevated FA and also susceptibility, indicating its highly organized microstructure and also iron content. But this is slightly different when we look at the cingulate cortex, which shows high FA but lower susceptibility. We also assessed the visibility of some cortical lamina structures, especially around the pre- and post-central regions, and also some in the posterior visual areas. So from that, we can see that the ex vivo QSM showed the best visibility of those lamina structures. But the in vivo QSM suffered a bit from the motion artifact, while the diffusion FA also showed some lamination structures, but with slightly lower SNR and less sharpness. And in terms of the comparison between ex vivo and in vivo, they showed moderate positive correlations, suggesting, at least in the ex vivo sample, we had the anisotropic microstructure and also the myelin and iron content preserved in the postmortem sample. We'll skip this. So this is like ongoing project. So in the future, we hope to further improve the acquisition, especially for the in vivo QSM STI with motion and field correction, and also improve our STI reconstruction and modeling, and then apply this to our future deep STI network training. I would like to thank all the members in our Hopkins group involved in this project, and especially Susie and Birkin and all the colleagues in the Martino Center to help us set up this project and help us run scans and all the funding resources. Thank you.